herzlich willkommen zu meinem ersten Video, I guess. Das ist hier mein Kanal Bio-Erklär. Ich versuche so ein bisschen Bio zu erklären, weil gerade so manche Sachen sind halt ein bisschen schwieriger zu verstehen und ja, ich will den anderen so ein bisschen helfen, deswegen erkläre ich das so. Ich werde das Intro immer auf Deutsch machen, um zu erklären, worum es im Moment so geht und was so das Thema ist, und dann werde ich auf Englisch switchen, weil ich habe Bio auf Englisch. Ja, genau. So, genau, also das heutige Thema ist praktisch Inheritance, also Vererbung und woraus wir eigentlich so bestehen. Also so Proteine und ja, DNA-Duplikation und so weiter. Also praktisch ja, Mitose und Meiose auf so einem tieferen Level, mehr oder weniger. Also, genau, ich hoffe, ihr versteht alles und ihr habt Spaß. So first off, we start with a human cell. It could be a normal somatic cell or also gamete. It doesn't matter. The fact that it has a nucleus is really important. Yeah, because inside of the nucleus there are chromosomes made of DNA tightly wrapped around histones. These are proteins. And yeah. So what you can see here, here's a chromosome. The DNA is wrapped around a histone here, and there's like an enlargement. It's not really happening, but just so you understand it better. There's like this DNA, and inside a enzyme, which is called polymerase. Remember, the ending ase is always an enzyme. It's like splits the DNA up, so we have two one-stranded DNAs inside of the polymerase. Then there's a process happens where a RNA, where mRNA, messenger RNA, is made, which is like sort of a copy or a copy, but an encoded copy of the one single-stranded DNA here. I made that green because it's important later on. It's called a mRNA because it's for messenger RNA. And as I said, it's the copy of the one single-stranded DNA but encoded. Okay, so now that the mRNA is like finished, so we have a full copy of a part of the DNA, it travels out of the nucleus into the cytoplasm, which is like here. So we're still in the cell, but not in the cell core anymore. What happens now is that there is another enzyme which doesn't end on ASE, so ACE, but it's still an enzyme. It's called a ribosome, and what happens here is that the mRNA, so the messenger RNA, is like connected or for a short time to a thing that is called tRNA molecules, so the transmitter RNA molecules, and they transmit a thing called the amino acid, which is then made into a chain. That was very much, so I'm going to explain it to you in detail. So the mRNA is a copy, but encoded copy of the DNA, and in the DNA things like the plan for how proteins are made and other things are written. So it's like a manual on how to build more of the things that you already have in your body. Now that we have a copy of this, there are some other things like the tRNA molecules that have little genomes, like these little toes, for example, and they attach to the mRNA in the order it should be, and then release the thing that they have. For example, you say on their head, the amino acid, a single one. There are 3 genomes read at a time, and then the amino acid is disconnected from these tRNA and connected to other amino acids. And as I said, the order is determined by the mRNA. Yeah. After this chain is finished, this amino acid chain transforms into a protein, so like a clot, to make this process to do this process all over again. So it could be that an amino acid chain forms into a protein, for example a histone, as I said, the DNA, what the DNA wraps around, or other things that we have in our body. So I talked lots about like corresponding and matching right now with the mRNA and the tRNA and these little toes, and that they have to match for the amino acid to get into this chain to make protein. But I'm going to explain to you how it is like in detail and what happens there and what I mean with matching or corresponding. So when you have 3 of these little toes are like nucleotides, either the mRNA or the tRNA, you have a codon. So like a group of 3 nucleotides is a codon, which corresponds to a specific amino acid or like stop signal during the process of protein synthesis. How do they correspond to each other? This is a pretty, like, you could say simple question, but in detail it is a bit more complicated, but don't worry, I'm going to explain it to you right away. So the most important thing to remember is these letter combinations. T corresponds with A and C corresponds with G, and the other way around. If we look at the double-stranded helix, the DNA, you'll probably remember that the one helix is going this way and the one helix is going this way, and they're connected by I also forgot. Nucleotides. Right, they're nucleotides. Yeah, and so they're so A stands for adenine, C guanine, C cytosine, and T thymine. That's like really important, and these are the nucleotides from the DNA. That's how the double-stranded, like the helix, connects to each other. Now we'll look at the RNA. The RNA is basically a single-stranded version of the DNA and is encoded. So it's basically a copy, but yeah, encoded, so a bit slightly changed. Instead of T, now we have U, because in the RNA the thymine doesn't exist, we only have uracil. But it also corresponds perfectly fine with the adenine, so there's no big problem. Just so you know why with RNA we use U and not T. From DNA to RNA, the transcription happens. Translation is later on with the proteins and the amino acids and so on. But there's a transcription, so we take the T, for example, and transcribe it to U because it's equivalent to the T. So now we have this wonderful wheel, which is called the CodonWheel. So we have 3 levels, the first one, the second one, and the third one. I didn't complete the third one because there was very much to do, and it doesn't really, well, it does matter if you look at it in detail, but you can just Google the CodonWheel and it should pop up. I'm gonna put a link into the description for a good CodonWheel. But for the explanation, I think this is sufficient. So we have, as I said, the first level, where the first toe or nucleotide is determined. So I'm just gonna, for explanation, do the mRNA here, where we have these 3. There's codon. This part is called a codon. 3 nucleotides. If we, for example, have the amino acid tryptophan, which consists of 3 nucleotides, it will be U, G, and G here too. And that's how you basically read it from the inside out. And that is basically it. So there's codon or amino acid tryptophan, tryptophan would be U, G, G. And yeah, that's like the first step. So now we have the mRNA and that stuff, but we still have the tRNA and the amino acids, so it's not fully done yet. We can leave it like this, but this is not the mRNA anymore, it's not the tRNA, just so you're not confused. So we figured that the mRNA is for tryptophan is UGG. So now we still have to remember, okay, we had that thing with the letters that match. So yeah, then we're going to just write the opposite for the tRNA, and that's it basically. So for U it was A, and for G it was C. So we have the exact like opposite. Yeah. So A, C, C. Right, you get it. And that's basically it. That's the tRNA for tryptophan. A, C, and C. So if you later then combine these two nucleotides, there's 6 nucleotides with each other. We have ACC on the side of the tRNA and the UGG on the mRNA side, and then they can bond. The combination I just mentioned is also called the complementary bonding. Complementary things you might know from like arts because they're complementary colors. For example, yellow and purple, and it's just like with this one you have a wheel, and almost on the exact opposite there is the corresponding part you need. Oh my god, but how do I know it's tryptophan and not serotonin or something? Well, usually on a CodonWheel it's written on the sides which letter and which nucleotide combination equates to which amino acid, and here on the sides it will for example be tryptophan, and usually there's also like short versions, for example S or TRP or something else for tryptophan, for example. But yeah, that's basically it. Alright, it's nice and stuff that we know how to get from mRNA to tRNA, but how do we get from DNA to mRNA anyways? That's very easy because it's the same like process that we just had right now. So you just have letter combination again, for example ACC, which is tryptophan, right? And you just translate it again. So the opposite of, so when you have here the DNA, what you remember is T to A and backwards, and C to G and backwards. So when you remember this, that we go from T to A and backwards, and from C to G and backwards, you could say, well, it could be TGG. That's wrong, because we're not switching between DNA parts, but we're switching to something else, which is the RNA. And in RNA, we have the different things, the different nucleotides, which are U and A corresponding, and C and G corresponding. So what would it be? You can pause the video or like write in the comments what you think what it is, or I don't know, get a sheet of paper and write it down. It's in the end UGG, which results in tryptophan, right? So yeah, that's how you came from DNA, boring, to a fucking protein. Wow. Well, not exactly protein, but almost there. Okay, so now that we had that, I'm gonna give you an example of what it could look like. Okay, so this is an example of what it could look like. And as we have a codon wheel, we use this because this is the original codon and this is the anti-codon. I think there are also anti-codon wheels, but we are working with the codon wheel right now. So from this, we could look into the codon wheel for ACC and we would find tryptophan. Yeah, tryptophan. I am so very happy to tell you that you've done it. You've now got how you can go from DNA to a protein to amino acid and then to a protein. And yeah, well, I hope you like this video, and if you have any questions, just write down below in the comments.